Well, here we are, starting what looks like 9 o'clock, almost right on the button, maybe a little bit late. Um, Good morning, everybody. That was Phil Collins that you just heard from the immortal group called Genesis. And it's kind of funny. The music today will be by Genesis, and the topic is genetics. So, anyway, I'm Dr. Minarsik, as you could see here from the chart, and it's August 27, Tuesday, 9 o'clock in the morning, Central or Chicago time. This is our 8th webinar in what will be about 60 or 70. In our online global pathology course, today's topic is genetics. We have a little bit of an improved studio today too. I was getting a lot of backlight, so we put kind of like a dark drop back there, and maybe we even improved the music a little bit, but I think we improved the video even more. And the reason why I said yikes about genetics diseases is you're going to find out pretty soon. Let's do our test. And by the way, our graphic theme is marbles. So let's do our AVM test to make sure everything is working good. I think it is, but let's get your opinion as well. Can you hear me? That's the A test for audio. Somebody has to type in the answer. Okay, how many fingers do I have up? That's the V for video. Okay, good. And if you hear me, that means you heard the music too. So on a scale of 1 to 10, what was the quality of the music that you heard from Phil Collins of Genesis? Oh, I'm getting a 9. Good. We got 7s before, so maybe we did tweak it a little bit. Maybe I just played it a little bit louder. Let's get down to business. We're not going to be in any big rush today. I think we'll be able to do everything nicely. I don't really want to make any more announcements, I don't think. But I do want to say something about our topic in general for a few minutes. This is a very special topic, and I normally don't take much time to talk about the topic itself. But we're going to talk about genetics today, or genetic diseases, okay? And the reason why I put all of these crazy comments, impossible, horror, anxiety, dread, inadequacy, explosion, and they put or easiest. I got to say something special about genetics and genetic diseases. If you feel like you are inadequate or dumb in genetics, I got news for you. I want you to join everybody else in the world. I don't know a single person, including genetic Nobel Prize winners. I met one who feels they really are up to date on genetics. You know, you might have gotten your A in your genetics course and loved it, and 2 years later you're practicing medicine and you realize you don't know anything. The field is exploding every year. And I can't keep up with it either. I'll be very honest with you. I noticed they added a brand new introductory chapter in the latest edition of Robbins about concentrating on genetics and molecular biology and stuff like that. But what I decided to do is go through all of the classical principles of genetic diseases. And I have given up trying to keep up with all the new discoveries of genes and diseases and treatments. Maybe I'll make mention to them a couple of times, but what we're going to talk about today is the classical genetic diseases and genetic principles. So, you know, we have always talked about different kinds of diseases, and one of the diseases you could say is genetic diseases, and you could also probably say that all diseases are genetic diseases because all diseases act upon or are the result of our genome. And if you wanted to throw in environmental factors as well, you can. So probably a very good true statement would be all diseases are genetic with environmental influences as well. You know, and people have debated, is it genes or is it environment? Well, it's both, of course. So Probably the single most important concept in genetics and genetic diseases is the concept of a mutation. I think you know what that means. It means an alteration, a change, maybe a damage to a gene or a chromosome. So we're going to be talking about mutations going from the gene all the way up to the whole chromosome. In fact, you could even go down to a change in a single base error, okay? It could be the substitution or a deletion of a single nucleotide at the very lowest level, many genetic diseases. Or you could talk about genes or perhaps even segments of a chromosome. A gene could be maybe 20, 30, 100, maybe a million base pairs. Chromosome, oh, I don't know, probably anywhere from 50 to maybe 150 million base pairs. You could talk about genome mutations or whole chromosomes, and that's what we're going to do. Even though I have it listed genome, chromosome, gene, we're going to barely talk about them from the bottom up. But this is one way to look at mutations, okay? Now, if you want to think of a gene as part of a chromosome, as many nucleotides or base pairs, at the very simplest level, you could think of it as a single deletion of a single base. You know, AGTC, you know, you could have one of those either as a deletion or a substitution, okay? That's probably the easiest way to think of it. In fact, the example I'm going to use is something that is always talked about as sickle cell disease as the classical disease involving a single base pair. You know, the hemoglobin molecule has alpha and beta chains. You might remember that. I'm sorry, beta chains. And in the beta chain, you have a trinucleotide. You have GAG. And that trinucleotide codes for glutamic acid, and glutamic acid is in the beta chain, okay? But if that single A from GAG is substituted with a U, now you have GUG. And of course, a different trinucleotide means a different amino acid. So now we have valine rather than glutamic acid as part of hemoglobin S rather than normal hemoglobin A. And that could be considered a single point mutation. It doesn't cause a shift in the whole frame. It doesn't, but it does code for a different amino acid. And this error is carried on from DNA to messenger RNA, all the way into translation in the production of the amino acid, and then the production of the polypeptide, and the production of the chain, and the production of the globin part of hemoglobin. And that one single thing makes the difference between a normal person or a person with sickle cell disease. In a gene mutation, in this case, we had a substitution of valine for glutamic acid, but you can also have mutations in parts of the genome or genes which do not code for proteins. In other words, they're regulatory portions of genes which are involved in transcription or regulation other than the production of proteins or polypeptides. You can also have what they call a frame shift mutation. Now, if you remember, in a simple substitution where you had one single point mutation and now the production of a different amino acid, what if the mutation was not or interrupted that multiple of 3? Then you have a whole shift of the DNA, don't you? And now you have many abnormal amino acids. Now, could those be carried over into a transcription as well? I really don't know, okay? Also, think of a record. You know, when you have a record, it's maybe many bytes or sound bytes long. Remember the old LP records or the 45s, what they call vinyl? Do you remember when something would happen in part of that circular spiral and there was a skip or a flaw? Well, the whole thing played over and over and over and over again. And that's that's the classical example of a gene mutation involving trinucleotide repeats, where you have certain repeats which could be repeated many times, also because of a mutation. Now, if you want to think of genes as producing proteins classically. You could say that not only can they interfere with the production or make an abnormal protein, but there could be a suppression of transcription now from DNA to messenger RNA. Or a abnormal messenger RNA could be produced from an abnormal DNA. Now that you have messenger RNA that's abnormal, this is carried over into the translation phase with transfer RNA. And in gene mutations, you now have abnormal proteins which may not at all interfere with the synthesis of proteins or even the synthetic rate, but just the synthesis of the proteins themselves. You're getting abnormal proteins because of abnormal amino acids, because of abnormal changes in the base pairs, ultimately. So when we talk about genetic disorders, here we have the same chart we more or less had before, but now this is going to be the order in which we'll be discussing them today. We'll be talking about single gene mutations, perhaps a single base pair. You know, genes can be many, many thousands of base pairs. I think 27,000 is about the average, but they could go up to a couple million, I think. And we'll be talking about the mutations which follow the classical Mendelian inheritance patterns, okay? We don't have to go into the P experiments with Gregor Mendel, but I think you know concepts like autosomal dominant, where one abnormal gene or allele is present, or autosomal recessive, in which you have two. And then we're going to move into what we would call either multifactorial inheritance, which involves more than one gene. And I guess you could say it's also multigenic as well, because it's multifactorial. But you have to remember, multifactorial is probably not the very best term, and multigenic is really probably not the best term either. But classically, diseases which are the result of more than one single gene could be called multifactorial, because, you know, environment is a big factor as well. Finally, we're going to go into parts of the chromosome more than just genes. We'll go into fragments of the chromosome, and we'll go into the whole chromosome itself, or we have chromosomal or perhaps genomic disorders, and that's how we're going to handle it. Just to refresh your memory, and I'm sure you knew this in probably high school or grade school, inheritable diseases can follow an autosomal dominant, an autosomal recessive, or a sex-linked pattern. They all have their own characteristic and pedigrees. An autosomal dominant disease is a disease that is the result of one abnormal, or we'll say mutated if you want, allele corresponding to a normal one. In the autosomal recessive, we have two impaired genes. It's double, okay? And in sex-linked, we have any mutation involving simply the X chromosome. So the sex-linked inheritance patterns are different from the recessive. They're different from the dominant. They follow different pedigrees. And in the sex-linked, we're only talking about problems on the X chromosome. And you could map out the X chromosome and, you know, talk about little segments of them. And when you look at those little segments, you could name, you know, the whole wide variety of X-linked diseases that are considered like in males only. So let's go to the classical first one, autosomal dominant diseases. The disease is spoken of as being heterozygous, or due to heterozygotes, okay? Now remember, they may have not inherited the abnormal allele from a parent. It could have resulted as a new mutation in the person as well. An autosomal dominant disease is something that has reduced penetrance or full expression of the disease, or full expression of the genetic problem. And the reason why it's reduced is there are environmental factors, or perhaps other genes may be involved. Now, closely related to reduced penetrance is something called variable expressivity. In other words, it may be severe, it may be mild, it may be absent, and another classical feature of an autosomal dominant disease, it may even be delayed. The person may never even know they have it until they're in their 30s, 40s, 50s, 60s, okay? And most of the autosomal dominant diseases and the mutations result in a reduced production or an inactive protein. Okay, so the proteins ultimately will either be reduced in production or inactive. Probably the single most two things to remember is that they have a variable expressivity and they could have a delayed onset in life, okay? And that pretty much is the way you could look at them clinically, because when we talk about autosomal recessive, it's basically the opposite. So, do I expect you to memorize all these diseases? Have you seen all of these diseases? Do you know what they are? Have you even seen some of these terms? I certainly do not have the time to go into them now, and maybe we'll talk about some of them. But during the course of this course, we are going to probably talk about almost all of these diseases in greater detail. What I kind of expect you to do, what I kind of expect you to know, is that if you see these diseases, and I'm sure you've heard of a lot of them before, you should know that they are autosomal dominant. So if you look at these diseases, with a couple of exceptions, okay, if you look at the diseases or you know somebody that had them, or you had a patient, or you have it, or somebody in your family has it, just remember that these diseases generally follow this pattern: delayed onset in life and variable expressivity. Okay, they're not uniformly expressed. It may be because the gene is generally reduced penetrance. And why might that be? Well, I'm not sure if I know the exact biochemical reason, but when you think about it, an autosomal dominant has only one defective allele. So maybe it's not as fully expressed or early expressed as something in which there are two defective alleles, like you have with the autosomal recessive. So those are the diseases. If I went into an explanation of each one, I would be not doing you a great service, because I'd be wasting your time the rest of the couple hours. This is a classical autosomal dominant degree, or pedigree, we'll say. It's both in males and females, and it does not skip generations. Why? Because all it takes is that one dominant gene to be carried into the next generation to make the disease. You don't need two alleles to do it. If you can't understand that, just understand that both sexes are involved. In other words, it's not sex-linked, and generations are not skipped. So let's go into the next type of pedigree. The next type of genetic pattern of diseases are the autosomal recessives. And here you have homozygous situation in which you have, let's say, two defective alleles, if you want to use that term. It's probably not biochemically correct perfectly, but it's the way of thinking. Now you have two defects pairing each other rather than only one. So it's going to result in a more complete penetrance. It usually occurs early in life. Almost all of the classical childhood genetic diseases are autosomal recessive. Many, perhaps even most of the autosomal dominant might not appear until sometimes even, late in life. It's a uniform expression. It's not so much a variation of mild, moderate, severe, okay? And it's not likely that new mutations are detected clinically like you have with the autosomal dominant. And now we have proteins which show loss of function rather than delayed or inactive, okay? And if you take the big family of what we call inborn errors of metabolism, many of them are enzymes, or they could generally more or less be grouped as the lysosomal storage diseases. These are basically all autosomal recessive. So now you know that this big family of inborn errors, use the word born, it means it's probably present when you're born, okay? That's an interesting parallel. They're much more common than the autosomal dominant, and it is the predominant type of genetic disease present in children, newborn people. Here's a bigger list. Okay, we'll be talking about these. Some of these we'll be talking about today in minor detail. But once again, I know this might be pushing you, and I'm not telling you you have to know this or you have to know that. But I will tell you, I think it would be really, really good if you sort of remembered these diseases, some of which you may have heard of and know because they're autosomal recessive. They appear early in life. Another easy, if you can't do that, then what you could do is probably make the general conclusion that any genetic disease that's present at birth is probably an autosomal recessive disease. That there's some exceptions to that, but that's another way of looking at it. Now, because you need two abnormal alleles, the chances of them getting together in the next generation is not as high as an autosomal dominant. So now we have generations that can be skipped as well. And because it occurs both in males and in females, you know it's not X-linked. That's the autosomal recessive pedigree. Generations are skipped. In the autosomal dominant, generations are not skipped. Okay, let's talk about the sex-linked diseases. These are diseases that are only linked to the X chromosome. So therefore, a person that has only an X chromosome, one, like a male, is not going to have the disease, right? Because there's nothing to pair. Only males can get it, right? Females cannot get sex-linked diseases. And I know what you're thinking. You're thinking, well, if a woman is XX, why can't she have a double dose? Well, it just doesn't happen. So it's only in males. And if he has a son, remember, he's only passing on the Y chromosome, so his sons don't get it. But all of his daughters are carriers, okay, because they get his X chromosome. Now remember, even though all of the chromosomes are regarded as homologous or have pairs, the Y chromosome is not homologous to the X chromosome. So technically the concept of dominant recessive have no meaning here. Now this last thing is something I still have a hard time understanding, because a lot of times you'll be reading about a disease and it'll say, oh, it's X-linked dominant or X-linked recessive. Can I just make a comment here that would, even though there is a way of understanding it that way, just think of dominant and recessive not having much of a meaning here. Okay, if it's X-linked, it's only in males, and you shouldn't be using the word dominant or recessive. These are classical male diseases. Now, you already probably know that only men get hemophilia, A and B, both types. The G6PD deficiency and enzyme deficiency only in males. The classical agammaglobulinemia and immune diseases in boys as well. Muscular dystrophy, Jerry's kids, the Duchenne, the Becker muscular dystrophy, they were only in males. So when you remember Jerry's kids, if you are old enough, they were always generally boys, okay? Another immune disease called Wiskott-Aldrich, diabetes insipidus, Lish-Nyhan syndrome, a severe form of gout, and another interesting disease we'll talk about at the end, the Fragile X syndrome, in which trinucleotides are repeated many, many hundreds or thousands of times, something that has opened the door to a whole bunch of trinucleotide repeat diseases. So here's the pedigree. It's very simple. It's only boys. And skipping generations doesn't really matter, okay? If you have a genetic disease that's only in males, you can bet the problem's on the X chromosome. And if you had an X chromosome in your hand and you looked at it, and you looked at the bands and you looked at the subbands and you looked at the arms, and you could actually put a little red dot where the problem was, you could probably map it out to the specific disease. And we're going to show you an example of that as well. So let's go back now that we learned about all the pedigrees and inheritance patterns and to the concept of a single gene disorder. Let's say that a single gene, for example, codes for a single protein. Let's make that assumption, although it's not exactly always true. Maybe at a polypeptide anyway. But if you have a gene that's mutated, it's going to produce a protein like an enzyme that is also not going to work very good or not work at all. And if an enzyme doesn't work, you have accumulation of your substrate, you have the lack of a product, and you have failure to inactivate a protein, which causes the damage, especially if it's building up inside of a cell. You could have a protein that's a receptor protein. For example, proteins have receptors, and the protein receptors are also proteins. So if a protein or any other substance cannot go to its receptor, like, for example, cholesterol, going to the receptor in the liver that transports it through the liver, then the cholesterol can't get into the liver for processing, and it builds up, and guess what? Maybe it takes another route and gets gobbled up by macrophages and forms plaques and cholesterol crystals, and you know the rest of the story. That's one of the most severe form of a familial atherosclerosis. Okay, now what many proteins are structural proteins, like collagen, like elastin. Okay, in fact, if you remember, I think it was a couple of webinars ago, we talked about the various types of abnormal collagen producing various types of diseases. So the now defect in protein can interfere with its structure, its function, or its quantity. And here's another interesting concept. An enzyme defect might result in a reaction to a drug, or let's say a drug working properly rather than, a reaction like a side effect. For example, male patients with the X-linked disease G6PD deficiency might have a problem with susceptibility to primaquine, which is anti-malarial. And that's probably the classic example. There's a bunch more, and I have a feeling you're going to learning about a bunch more in pharmacology as well. So what are the structural proteins? Well, collagen, obviously, is the biggest one, but we also have something called elastin, okay? And elastin is made from fibrillin, okay? And fibrillin has 3 types, but when you have a fibrillin 1 defect, which is the main one, you now have a defect with elastin. Hey, do you remember all of those elastic fibers in the aorta? What would happen if your aorta or your aortic arch no longer had proper elastin and it couldn't be elastic enough or rubbery enough? Hey, well, maybe it would be degenerate and develop aneurysms. Well, that's one of the big problems with Marfan syndrome. They also are very tall, and they have a variety of other things. A lot of people say Abraham Lincoln was a Marfan syndrome, tall guy. We don't know whether he had an aortic aneurysm or not. Osama bin Laden, one of our students has Marfan syndrome, and he just happens to have had heart surgery already or aortic surgery, but he also happens to be probably the smartest student I had a couple of years ago. Ehlers-Danlos syndrome. I'm glad that there's an S there because it's not one genetic disease, it's several of them. But the thing that they all share in common is the fact that they have abnormal collagen. So as we have abnormal elastin in Marfan syndrome, now we have abnormal collagen in Ehlers-Danlos or ED. There's autosomal dominant and autosomal recessive forms. And there could be at least 3 different types of abnormal collagen. Okay, and actually they said there's 6 different types, and they're given different adjectives. But forget about that. Think about what would happen if your structural protein collagen, which is nice and firm, all of a sudden was defective and it couldn't be firm anymore. Well, maybe the tissue that it supported or gave strength to would lose strength. Maybe your joints would become more mobile. Maybe your skin would stretch more, okay? And I have a very, very good, famous friend, probably really, I'm not kidding you, the world's most famous rheumatologist who gave a lecture to our students once, and he asked, how many of you are famous athletes? How many of you are famous dancers? How many of you are really good at acrobats? And he says, chances are 50% you probably have some type of abnormal collagen, which in many cases doesn't result in a disease, but it might result in the person who has it getting a Nobel Prize, for some type of sports activity. Okay. Let's go on to the next concept of a receptor protein defect now. The LDL receptor is a protein. If it's abnormal, it can't receive the cholesterol for transporting across the liver cell, which is the main pathway for the processing of cholesterol. So what happens? Well, the cholesterol then builds up in the blood. If it can't be cleared by the liver, what is another way to clear cholesterol? Well, remember, the macrophages can eat it, and not only can they eat it, but they can deposit it through the endothelium into the arteries. And now, like I said here, you know the rest of the story. So there is a familial hypercholesterolemia, which is a genetic problem involving an LDL receptor defect. It's a buildup of macrophages, and of course, with the cholesterol building up even more, perhaps cholesterol crystals or cholesterol ester crystals, more chemically correct. And that could be an advanced. People with familial hypercholesterolemia very often die of atherosclerosis effects early in life. Okay, well, everybody knows what enzymes are. We all know that they're proteins, and we know that they take a substrate and they change it into a product. That's what an enzyme does. Well, what happens if it can't because it's defective, because the gene was defective that codes for it? Now we have a buildup of substrate. Buildup of substrate cannot be degraded very often, and therefore it's harmful. It interferes with the cells, it interferes with the tissues, it interferes with the organs. Now, most of the diseases, classical diseases, which we call enzyme deficiencies, can be lumped into the category of lysosomal storage diseases, because lysosomes are a big bag of enzymes processing and degrading just about every type of chemical known to man. So if these big bag of enzymes have, if any one of them is defective, it could result in a buildup of the substrate in the lysosome. So the big category, which I'm going to go through in a little while, the lysosomal storage diseases, is also enzyme deficiencies. You know that these are primarily autosomal recessive as well. Most of the classical enzyme storage or lysosomal storage disease appear early in life, are in kids. Okay. One of the lysosomal storage diseases are diseases in enzymes which process glycogen. So if glycogen cannot be processed into glucose, the glycogen builds up, okay? All of these have the same general logical principle. In the sphingolipidoses, the gangliosides build up because the sphingolipidoses cannot be processed by the proper enzymes. In the sulfatidoses, sulfatides, there's a couple of main ones, but this general family of chemicals cannot be processed by enzymes, and therefore the sulfatides build up wherever sulfatides would normally build up. In the mucopolysaccharides, okay, if you have a deficiency of mucopolysaccharide enzymes, the mucopolysaccharides would build up. And they're technically different from the mucolipidoses because this is a protein and a carbohydrate moiety, and this is a protein and a lipid moiety. But the same general principle. If they can't be processed by their enzymes, they build up. And if that wasn't enough to blow your mind, I got news for you. I'm going to do a brief mention as well about fucosidosis, mannosidosis, aspartoglycosiminuria, Wolman's disease, acid phosphate. These are all things which can build up because the enzyme which processes them is defective, and they all generally occur in this area or milieu of the lysosome. Well, where does the body store glycogen the most? Well, it stores it where it needs it the most. Skeletal muscle, liver, cardiac muscle. These are things which are glycogen magnets. And so that's where normally you have a lot of glycogen activity. If you looked at a heart or a liver or a skeletal muscle, you'll see these tiny little black dots on electron microscopy. That's glycogen. Well, what happens if you have enzymes which can't process them, degrade them into glucose? Well, then you get a glycogen storage disease, and these are the primary areas for glycogen storage diseases. There's 10 types. You may hear the name Pompe, von Gierke, McArdle. These are the classical first 2 or 3 that have always been discussed. Now there's a whole bunch of them. I put down 10, and this was like about, a few years ago. I wouldn't be surprised if there's a 12 or 13 classical ones by now. So that's the concept of a glycogen storage diseases. They all have different clinical presentations. Some could result in a cardiomyopathy because you have more glycogen in the heart than you do have functioning muscle fibers. Some of them are builds up in liver, could cause hepatomegaly. Some of them are multiple. And if you went, I had a list of about all 10 of them in their different presentations and their different names, but I'm not going to torture you with that. You have to understand the principle, and that's probably the easiest principle. Now, here's another easy principle. A sphingolipid, a ganglioside, is a classical type of sphingolipid. It's a substance which is in a neuron. And I found out a couple years ago that the reason why it's called sphingo, that when it was first discovered, it was very mysterious. So they thought, well, it's mysterious, so let's name it after the Sphinx. How do you like that? Well, if gangliosides, which are normally present in neurons, are processed by an enzyme called hexosaminidase A, then they build up. And what could happen? Well, there's neurons can be damaged. They could die, okay? Now, the Ashkenazi Jews have a very, very high incidence of them. As many as 1 in 10 are carriers because it's a glycogen storage disease. You know, it's an autosomal recessive, so just being a carrier is not going to give you the disease. The central nervous system neurons are the primary site of accumulation. And one of the clinical things, here's a neuron that's just filled with ganglioside. You know, my son is an ophthalmologist and a retinal surgeon, and I asked him if he's ever seen the cherry red spot with Tay-Sachs disease, and I thought I would stump him, and he goes, yeah, I've seen him a lot of times. How do you like that? The sulfatides are also classical organic chemicals, okay? They include the cerebrosides, the sphingomyelin as well. These are things that are present in macrophages, in the production of myelin as well. Many of the sulfatidoses result in adequate function or production of myelin, like the metachromatic leukodystrophy. And you know leukodystrophy is a myelin disease because leuko means white and myelin is white matter. So you get it in the brain. You could get them in various other organs, like macrophages. But I think if you want to just look at the whole thing, you say a sulfatide could be an entity unto itself, but the cerebrosides are also chemically sulfatides. Sphingomyelin, myelin, is also a sulfatide. And I think I'll give you some example over here. Here's Niemann-Pick disease. It's, I think we have something here. It's probably one of the most common types of sulfatidosis. And if you had an enzyme which processed sphingomyelin and it was defective, it would then build up. This is also, unfortunately, a disease of Ashkenazi Jews' life. Like Tay-Sachs, it is very often fatal early in life because of severe CNS and organomegaly. Well, why would it be organomegaly? Because maybe it also builds up in the macrophages of the liver and the macrophages of the spleen as well. But of course, the most severe thing is the buildup in nerve cell axons as well. That's Niemann pick disease. Gaucher's disease, also very common, okay? Cerebrosides, okay? They're also sulfatides, or if you want to call them more chemically, glycosphingolipids. You can see them in muscle, you can see them in nerve, you can see them in macrophages, okay? And it's listed as the most common type of lysosomal storage disease. That's why I'm singling it out. And if you have an enzyme called glucocerebrosidase, and it's defective, then or deficient, then you know you're going to have a buildup of the glucocerebrosides. Okay, and these are also can be seen in some forms of it in central nervous system. Most of them do not involve the central nervous system. You see it in the macrophages, so it's a lot of buildup. You can have big liver, big spleen, and even it's one of the diseases that the hematologists love to study because you can also see these big foamy macrophages in the bone marrow as well. And I can remember looking at bone marrows with a very prominent hematopathologist, and he didn't know anything at all about the patient, but he saw these big foamy macrophages, and he goes, hey, I wonder if this person has Gaucher's disease. So yeah, he went upstairs, he looked through the chart, and sure enough, the patient had Gaucher's, or if they didn't, he told them that they did, and that he was correct. Okay. Let's go through another page of our biochemistry book here and talk about the mucopolysaccharides. Well, you know the muco means protein and the polysaccharide means carbohydrate. So these family of compounds, of which there are many, but we'll talk about two in particular because they are the most common type of lysosomal storage disease. Dermatan sulfate and heparan sulfate is a buildup because you do not have the proper enzymes for processing these. Now, classically, there are a whole big family of mucopolysaccharidoses, but the two most common and the two most classical and the first two that were discovered were Hunter's disease, which was number 2, and Hurler's disease, which is number 1. And I always have a hard time remembering that because Hurler is alphabetically before Hunter. I'm sorry, Hunter is alphabetically before Hurler, but Hunter is number 2 and Hurler is number 1. So it's a reverse alphabetical order. These are kids that are born with coarse facial features, a mental retardation, stiffness of the joints, and also because of the buildup of these sulfates, remember, they can get urinary excretion of them as well. Well, are you sick of the lysosomal storage diseases? I hope you are, because I just want to say a word quickly about some of the other families of lysosomal storage diseases. And by now, you could probably figure them all out, because if you have a fucosidosis, you probably know it's a buildup of fucose, okay? Because it doesn't have the enzyme fucosidase. Whoops, I better put a little space in there. Or if you have a buildup of mannose, another carbohydrate, maybe you have a problem with the degradation of mannose, so now you have mannosidosis, okay? There's a disease called Wolman's disease, okay? It has another name. There has a bunch of names. Acid lipase deficiency, in which you have a buildup of cholesterol and triglycerides. And you would probably think that if you had a disease called, which resulted in the deficiency of acid phosphate, you would have buildup of acid phosphatase. Okay, I know I took you through a lot. I'm almost thinking of taking a break right now. In fact, we will, okay, because we're finished with this big concept of the most common type of genetic autosomal recessive, this big family of lysosomal storage diseases. So let's take our 10-minute break. I'll see if I could get Woofy to open his eyes, that lazy dog. And I'll also refilter some of my coffee. And let's listen to 3 more beautiful songs by Phil Collins and his group called Genesis.